Well, hello everyone. It's my great pleasure to have this opportunity to talk to you all about CG9650, a Drosophila melanogaster gene that hopefully will permanently change the way you think about fruit flies. Defects in the proper formation and maintenance of mussels affect millions of individuals across the planet, with nearly 11 million people suffering from various forms of muscular dystrophy alone. In nearly all such cases, the affliction results in severe morbidity and ultimately a premature death. To gain insight into the biological mechanisms responsible for these developmental defects, we turn to Drosophila. Drosophila melanogaster, the common fruit fly, has approximately 60% of its genes present within Homo sapiens, the common human. Subsequently, an astonishing number of biological functions are conserved between the two species. And it's due to this homology that we look at myogenesis in Drosophila. This process begins, like many others, when a couple of flies fall in love. After this occurs, an embryo is produced, and about 22 hours after being deposited, this bundle of cells develops into the animated larva of everyone's favorite arthropod. 4 hours into this process, the progenitor cells for a variety of unique tissues become distinct. These cells will continue to replicate themselves until meeting the requirements necessary to cease mitotic proliferation and assume more specific roles, ending ultimately in the formation of their terminal structures. Currently, a model for this type of cell cycle regulation exists. The mammalian BCL11 genes have been shown to be involved in regulating the cell cycle through the active repression of another gene, P21, which initiates cell cycle arrest, leading to differentiation. CG9650 is, as we see in this protein blast, the Drosophila homologue of BCL11B, with the greatest conservation in amino acids seen in the region highlighted in green. Let's see. With this conservation apparent, we needed to address the question of whether or not CG9650 is involved in muscle development. And to do this, I turned to in situ hybridization of wild-type embryos for CG9650 RNA. What we saw when looking at the expression pattern in these embryos was a presence of the gene in the stage 11 embryos in the somatic muscle precursors, and then an absence of activity of the gene later on at the same site in stage 14 embryos. What this expression tells us is that the gene is being toggled on and off at the right time in the right location. And additionally, we observed that CG9650 is active on third instar larval imaginal discs, indicating an involvement in both larval and adult myogenic processes. And seeing that CG9650 is present in the developing muscles, we next needed to ask ourselves whether or not the mammalian relationship between BCL11B and P21 was intact within Drosophila. To do this, we wanted to see if CG9650 was acting upon DACAPO, or DAP, a cyclin-dependent kinase inhibitor similar to P21. And for this, we overexpressed CG9650 with crouple, a gene responsible for the proper segmentation of a well-defined region of the embryo, seen in this box. And then we performed in situ hybridization of these embryos with DAP RNA. What we saw then was within the domain of crouple, there is significantly reduced expression of DAP, indicating that CG9650 is in fact acting as a negative regulator of the gene. And so with this relationship now in place, we had to address the question of whether or not CG9650 was influential on muscle formation. Overexpressing CG9650 throughout the mesoderm, we fluorescently stained these embryos with tropomyosin in green, indicating the muscle fibers, and MEF2, a nuclear muscle marker, in red, showing the organization of the myoblast nuclei. What we saw in the mutants was severely irregular muscle formation and disorganization of the myoblasts. And to better understand these results, we had to distinguish which gene, CG9650 or DAP, was most directly responsible for the phenotypic changes. To do this, we looked at a line of flies carrying a loss of function allele for DAP. And by looking at these flies, we were able to simulate the repression that we believed was being facilitated by CG9650. And so staining, as we previously had, we observed similar lethal muscle malformation as well as disorganization of the myoblasts. And comparing these stains to those of the CG9650 overexpression flies, there's a striking phenotypic community seen. And so while this answers one question, it immediately begs others, and primarily on what level is the DAP interacting with the myoblasts. To do this, we turned to a LAC-Z reporter line for an enhancer of DAP. And so what we did was we took an enhancer of DAP and combined it with the LAC-Z gene and integrated that into a line of flies. During translation, the LAC-Z gene becomes active alongside the enhancer, thus producing beta-galactoside, which can be easily visualized and illustrates the activity of the enhancer. And so staining the beta-gal in green and MEF2 again in red, we observed that there was a significant amount of overlap between the enhancer and the myoblasts, indicating that DAP is active in a number of these myoblasts. And so with that, we can form a number of conclusions about CG9650 within the context of myogenesis. The mammalian model appears to be conserved within Drosophila. CG9650 appears to have a very influential role in the proper progression of myogenesis. This occurs through the active suppression of at least one target gene, which we are, of course, expanding our knowledge of as well. And so with that, I'd like to offer my greatest gratitude to my many friends in the CRIPS laboratory, IMSD and Flybase, you, my lovely audience, and of course, Drosophila. And I'm now pleased to take any questions you may have. Yes.
So, what kind of future work are you looking at now that you've presented them?
Well, I'm pleased that you asked that question, person I don't know. We actually have a number of really interesting experiments ahead of us, fortunately. To begin with, we want to gather some quantitative data on the relationship between DAP and CG9650. And so for that, we'll be looking at cell culture. Basically what we'll do is with our DAP-LAC-Z construct, we will activate it with TWIST, which is a very important transcription factor for Drosophila, and thus producing a base level of beta-gal. And then with the introduction of CG9650, we anticipate that this level will be decreased. We also are really interested in integrating our reporter line into our overexpression scheme to see exactly which myoblasts are being influenced. Easily the most important experiment is creating a knockout of CG9650, which is proving to be a challenging exercise, but I'm confident that we'll get there. Yeah. Yes.
So why has been creating the knockout been so challenging?
It's a good question. One of the best methods for reducing the expression of a gene is with the use of RNAi lines, and I'm not sure how intact that is within other model organisms, but basically you degrade the RNA of a gene before it's able to be used. And since the process we're looking at occurs so early on, we don't think that the RNAi have enough time to build up and be effective. Yeah. But we're doing a Pielman excision, which essentially is taking out a chunk of the gene, and hopefully that will produce the effects we're looking for. Yeah. Any more? Thank you.